Lauren Kirkland, and today I will be doing our fourth video demonstration of my microbiology lab class. And today we are going to talk about how to do a quadrant streak and a zigzag streak with our plates here. And before we get started, I'm going to review my lab attire, which I have my hair pulled back. I have my glasses on that are serving as my goggles or safety shields. I have on my lab coat, I have on long pants, and I have on closed-toed shoes. I also have made sure not to have on any excessive jewelry. Now, as always, I want to make sure I start any lab experiment with the proper hygiene and a clean working area, so I have made sure to do that. And now we are going to review the materials needed for this experiment. So first, I have here this blue inoculation hoop. Then I have a plate of our culture. Then I have two of my plates here that I'll be performing the streaks on, as well as my Bunsen burner. And then I also have a paper towel here that will help me sterilize my hoop in between streaks. And also, I don't have a pair of gloves here at home, so you would see gloves needed in the items list. So I will put on my gloves and we will get started. So first thing, we're going to do the quadrant streak demonstration first. So first, I want to make sure I turn on my Bunsen burner. I would light it or ignite it. And then remember, the vents at the bottom are controlling the temperature, and I want to look for the inner blue flame. The inner blue flame lets me know that's the temperature that will sterilize what I need to be sterilized. So I will grab my hoop. Now that I know my Bunsen burner is hot and ready, I will flame it, sterilize it. Okay. Now after this is sterilized, I would remove the lid, the top of my culture, and to make sure my inoculation hoop is not too hot, I would just barely touch the agar and make sure if it doesn't melt or melt, then I know it's cool enough. So I'm going to say it is cool enough. So I'm going to get a sample here of my specimen. Okay, put the lid back on. And then now I'm going to perform the first streak. So I'm going to slightly lift up my lid. And I'm going to streak specimen. Okay, put the lid back on. And then it's very important you sterilize in between your streaks, as you want different amounts of it in each streaks, meaning less in each streaks. So then I will flame it. Then I'm going to use this napkin here to serve as another sterilization technique. So now to do the second streak, I am going to turn the dish a quarter, lift the lid, and do streak 2, mixing in a little of streak 1. And before I did that streak, it's very important that I made sure to touch the outside to make sure that my inoculation hoop was not too hot. So again, I would flame it. Sterilize some more. Turn again for my third streak. Again, these streaks should be less and less each time. So I'm going to make sure my solution is not melting. It's not, so my hoop is cool enough. And I'll perform the third streak, getting a little of the streak two, mixing in with it. Sterilize again. And I'll turn one last time for my fourth streak. Lift the lid. Make sure loop is cool enough, which it is, and then I will streak the fourth time. Okay. Then I would make sure to flame, sterilize that. I'm going to set it right over here. And we are done with our quadrant streak. So I'm going to move this right over here. Now we're going to do a zigzag streak demonstration. So again, my Bunsen burner is still hot and ready, just like the last time before. So I would make sure to flame, sterilize my hoop. Take the lid off, culture, make sure my hoop is at the right temperature. It's not too hot, it's cool. Get a sample of the specimen. Put the lid back on. Then I will lift the lid, form a zigzag motion. Put the lid on, then I'll sterilize. Now in the zigzag streak, I do not have to sterilize my hoop in between each of the streaks because it is not specific to lesser and lesser amounts, like the quadrant streak. So after that is sterilized, I'm done with my zigzag streak. So here, again, before I close, I would want to make sure that I turn my Bunsen burner off before leaving the lab. I would make sure my lab area is clean, as well as good hygiene of disposing my gloves and washing my hands. And again, we went over today the quadrant streak of a plate as well as the zigzag streak. So that concludes Lab 4 and Video 4, and thanks for joining.